key up there as well, so have a look at the key and see what you got wrong and why you did. It's always something I recommend for people to do. You know, once in a while there's a mistake in adding or scoring, so if that's the case, just talk to me. But above and beyond that, you want to think about, you know, the scope of how you studied and how effective was it, and maybe how you can improve in the future. So that's there for you. I'll be handing these back probably for a break for you to look at. So with today's lecture, we're going to shift gear a little bit. We've been talking about some of these early landmark events: fertilization, cleavage, gastrulation, and variolation, in a variety of different species. And now we're going to take it back to mammals and look at these events in mammals. So I've purposely separated out all the sea urchin stuff with fertilization in the mammal stuff so that you can have a better chance to not get these confused. Okay, so one of the themes for today will be gametogenesis, and this is the making of gametes. And even though these are only haploid cells, they themselves have to go through a number of preparatory steps to be in the right condition and to be able to fertilize the right species once they encounter each other. So this is really definitely within the realm of developmental biology and an important part of the very first step in. So from this slide you can notice the morphology of the sperm versus the egg. And one remarkable immediate thing is that they're very different in size. And now that we've gotten to this point, and of course I think you can appreciate that there's some functional reasons for this. You know, the sperm, they have to swim, and we'll find out in females quite some distance to get to the egg. And then the egg is going to be going through this series of rapid divisions without pausing to grow in between. So it does make sense to have this as a deliberately large starter cell at the onset of fertilization. Mammals, like some of the other species we've talked about, do have these cells known as primordial germ cells. And these are stem cells that are going to divide many times to provide the cohort of gametes through the, either throughout the life of the organism or during the time that the gametes are being made. So a few features about them. They're set aside early in development, so it's kind of planned out by the embryo, seemingly which cells are going to be the ones that would become these stem cells. In many cases, it's a little surprising, but they actually have to migrate from the position that they arise in the embryo when they're first specified and determined. They're going to migrate through the embryo to find the gonad and then eventually take up residence there. So, another one of these surprises, you would think that they would just be born in the gonad and that's where they come from, but that doesn't have to be the case. And then as stem cells, they're first going to go through many rounds of mitoses. So just dividing themselves to create a population of diploid cells, and then those will go through meioses to create the gamete cells, the sperm or the egg. So for comparative purposes, I threw in this slide. So when we talked about Drosophila, we also saw a group of cells that were set aside really early in development, the pole cells. And those are the ones that would become the source of germ cells for the Drosophila embryo. They happen to be the very first ones that completed that process of cellularization and got membranes surrounding cytoplasm throughout the entire cell. Then our worms here, C. elegans, they have also specified the germ cell lineage really early in development. And those are the cells that happen to inherit these P granules that can be identified by our P granule antibody here. And so these cells have made that decision to take on germ cell fate really early in development. So we may see the term germplasm here, and that's just referring to this group of cells that will become the primordial germ cells or the cytoplasm that will become germ cells. So how does this happen in mammals? So So in C. elegans, we have a factor, this cytoplasmic factor, this morphogenetic determinant that seems to maybe cause or at least be associated with the germ cell phase. Is this a similar situation in mammals? And so far, we've not found a factor that seems to be positive or co-associate. Instead, what seems to happen is more of a cell-cell interaction event. So an induction event where maybe one set of cells is sending a signal and the other set of cells is receiving that signal and being influenced by it. And so this is what the mammalian embryo looks like early on. And we're not going to go through a great deal of detail on what the layers are and where they came from because it is rather complicated. So I'm just kind of jumping ahead from the stage when we're at the blastocyst. So we have this group of cells that has divided and become shaped like a soccer ball. And then it's going to kind of sort out into a layer. And then a little bit later, at about 7.5 days of development, we have a layer of cells here, another layer of cells here shown in green, and then another layer up there shown in purple. And two of these layers will eventually contribute to membranes that surround the embryo and become part of the placenta. So any of those cells would not be part of the embryo itself. So they're extra embryonic. Then there's one region of the embryo known as the epiblast, and that's shown here in blue, and these are the cells that will become all the different daughter cells encompassed in the embryo. And the primordial germ cells shown in red on this slide, these are developing right at that corner at the interface between these two tissues. Right here, and there's even a mouse line where the primordial germ cells are marked with GMP. So, what we see is these extra embryonic cells, they start releasing BMP, which is bone morphogenetic protein. I don't think we've actually, that's one of the TGF-beta family members. We haven't talked about it in too much detail. But this is a paracrine factor, and so when it's released by these extra embryonic tissues, it can migrate down a few cell layers away and then induce the germ cell fate within these epiblast cells. So if these epiblast cells are competent, that is, if they're prepared in molecular terms, then they'll be able to respond to this signal that they receive. And you can see some changes in gene expression that indicate that response, and these include expression of certain transcription factors like the BLIMP or the PDRM4. And this is the first steps towards those cells saying, okay, I have information to be specified as germ cell fates, and then this process of commitment will continue with them. So this is the first time we've talked about the idea of competence. And not all cells would necessarily be able to respond to an inductive signal. They really have to be sometimes prepared in terms of their lineage or their location or their own set of genes that they're expressing to be able to respond to this. So for example, these cells might be the ones that have a receptor for BMP that they're currently expressing, whereas other cells of the epiblast may not be expressing that receptor. So even if they did, manage to get some BMP, they're not going to be able to respond to that inductive signal and become germ cells. So there's kind of two parts to this. Yes?
Sorry, with that, did you say they need to be expressing the BLIMP one and another one to be able to get the inducing signal from BMP, or the inducing signal makes them express those other two?
The latter. So once they get the signal and they are responding and taking on the induced state, you'll see the expression of some downstream genes like BLIMP1 and PDRM4. But then in addition to that, these cells at an earlier time probably already were doing something special to be competent to receive that signal. So induction and competence. Then these cells are going to start to migrate. And so here you see that over on the right-hand side, the PGCs, these are the primordial germ cells. and they are going to migrate down through the epiblast portion of the embryo. So this is a layer, it's a couple layers thick, this epiblast, and these germ cells migrate as individuals. So they go kind of together in a clump, but they literally move down and in between all these other cells, and eventually they're going to take up residence at about this location here, in the region that will become the gonad of the embryo. And so people have been kind of interested in this, to know how it is that these cells, we can understand that they might be induced and that they're competent, but how do they know where to go and how do they actually carry out this process of migration? And it seems that one of the important factors is that there's an additional group of cells that migrate along with the primordial germ cells. And these are secreting a factor called stem cell factor, abbreviated SCF, I believe. So those cells plus the primordial germ cells kind of migrate together, and the presence of this stem cell factor seems to be needed for the primordial germ cells to stay alive and to keep migrating and eventually to get to the right place. So this is sort of an unusual situation where you see two different types of cells or two different self-baits of cells migrating together to get to a final destination. And I wanted to show you a couple of movies of this. I'll come back to that after the break and show you the movies. Okay, so they're migrating through this really interior region here. So this shows you the mammalian embryo with that big bulge up at the head, top being the head, and then we have the dorsal side over here, the belly side here, and this is the neural tube and the notochord. And then right here, these red dots are the primordial germ cells, and they're going to go quite a distance as the embryo continues to develop. And then eventually, when they get to the right anterior posterior position, they'll leave this hindgut area and migrate out into these two genital ridges, which are these two sort of bumps on the midline portion of the embryo that eventually will become the ovary or the testes. So looking at those genital ridges in a little bit more detail here, the hindgut is basically endoderm tissue that's going right down the midline of the embryo, and these cells are migrating in that portion. And then when they get to the correct anterior posterior position, they'll leave that and go out into these two genital ridges here and colonize within those cells. So they'll survive there, they'll continue to divide, and that's where they'll be for the rest of their life. So later on, we're going to have a lecture just talking about stem cells and the different types: adult stem cells, embryonic stem cells, and induced pluripotent stem cells. But for right now, I just want to point out that these primordial germ cells are highly pluripotent cells. So they are going to have the ability, if you were to, say, remove them from an embryo and culture them in the dish, they have a really powerful ability to differentiate into a lot of different downstream cell types. So they've been a tissue of interest from that point of view too. Okay. This slide I'm not going to talk about much, but I'm just sticking it in here so that I can come back to it later in some future lecture. And it's to point out that the gonad tissue is going to be developing right next to the kidney tissue. And eventually we'll find out down the line when we start talking about mesoderm and different portions of mesoderm that there's a territory called the intermediate mesoderm. And both of these tissues, the gonad and the kidney, develop together from the intermediate mesoderm. So it's a little bit of a preview for you. Okay, so what about the next step then of going ahead and making the sperm and the egg? And we're going to talk about these two processes in some detail. And I wanted to start out by just emphasizing that really, if you look at the bigger picture for this, there's some similarities that you see in the process for each type of gamete. So in both cases, we're going to find that they start from a primordial germ cell. And today is one of these lectures where your terms list is like that, because there's all these different names and words that are associated with this process of gametogenesis. So the first one here, spermatogonia, that's the name of the primordial germ cells that are going to make the sperm or spermatozoa. Then we have oogonia, that's how you pronounce that. Oogonia are the germ cells that will make the eggs down the line. And in both cases, the general process is pretty much the same. So the primordial germ cells, they're the stem cell-like situation. They're going to go through a number of mitoses until we finally get to a cell that we call the primary spermatocyte or the primary oocyte. That's a diploid cell, and that guy is going to now go through meiosis to make the haploid sperm or egg. Okay? So it gets a special name because it's the one that is poised to go into meiosis and make the sperm or egg. Then in general terms, spermatogenesis is the creation of sperm, or ovogenesis is the production of eggs. Question, yeah?
Yeah, when do those PGC cells become determined then? Is it in the primary spermocyte? 'Cause you said before that they can be turned into anything? So
when do the primordial germ cells become determined? So they are pluripotent, and I would say that they're already determined as primordial germ cells. I mean, so in normal cases, they're going to continue to go ahead and make these germ cells down the line. And
so they're determined and pluripotent?
it's kind of a little bit of a squishy definition because they're on their way to making primary spermatocytes, and you can hardly get more determined than that because they're just about to get to the next stage of being differentiated, if you consider primary spermatocyte to have some
so that'd be a
differentiated state.
Okay.
And yet, if you do take these out of the embryo, you can do manipulations to them. Yeah, I don't know quite how to answer that.
But by the spermatocyte, they're differentiated then?
Yeah, I would say that could be considered differentiated. I mean, they still have some, way to go before they become a fully functional sperm or egg. So this process of differentiation is going to continue. And you might look at it as a continuum of, getting more determined and getting more differentiated somewhat in overlapping periods of time. Okay, so let's look first at spermatogenesis. And so starting then with primary spermatocytes that have been derived from the stem cells known as spermatogonia, these are now going to start to go through rounds of mitoses, and they do so in synchrony. And interestingly enough, they also retain a little bit of a cytoplasmic connection from one cell to the next, and that might actually help them divide in synchrony here. You don't need to know all these little A1, A2, A3 business, but after several rounds of mitoses now, we're going to get to the primary spermatocytes, as we just said, and once again, these are diploid cells. Continuing on, these primary spermatocytes that then are going to head into meiosis, and so I'm sure you guys are all experts at meiosis, and we have meiosis 1 and meiosis 2. So we give some names to those guys also. After we go through meiosis 1, now we call them the secondary spermatocytes. So we've separated the homologous chromosomes here. Then in meiosis 2, we're going to become haploid, and these cells are now called the spermatids. And at this point, you're going to see some changes in the way that these cells look. So they go from being your basic round cell with cytoplasmic nucleus to being a highly specialized cell that we know and love as the sperm cells. So then we have a specialized word here too, another sperm word. Spermiogenesis is the process of converting the spermatids to mature sperm cells. And there are a number of different events that happen along the way to do that. And one of the things is that here you can see all these purple blobs. This represents most of the cytoplasm that those cells, those secondary spermatocytes had. They're getting rid of it because they want to become a very streamlined, jam-packed, specialized sperm cell. And so they don't really need or want all this extra cytoplasm, and it's actually sloughed off as a, quote, residual body that then just degrades because it no longer has any nucleus or anything. So sperm cells that are haploid, we can also call them spermatozoa or spermatozoon. So you're going to see some of these Latin endings. Generally the A is the plural version and the O is the singular version. Okay, let's revisit this, but thinking about the ploidy of the cell as we go along here. So the primary spermatocytes, they're diploid. They have 2 copies of each chromosome. So they have a mom chromosome and a dad chromosome, and after that goes through S phase, that will be replicated. So now we actually have, if we wanted to count up, 4 copies or 4 sister chromatids all there, as you see here. And then the homologous chromosomes are separating in meiosis 1, so yellow is dad's and blue is mom's, and there's any independent assortment for any particular chromosome you want to look at. So then we have these cells going through meiosis 2 where the sister chromatids separate. And so the copy number gets reduced, and also these are haploid cells at this point. Okay. So that's probably a review, but it's kind of good to have it all laid out. Okay, the male reproductive system. So all of this spermatogenesis is of course happening in the testes itself. And then eventually the sperm in mating are going to need to be passed through the male reproductive tract to the female. And so here are the testes, and there's a region known as the epididymis, which is a tubular region, and the mature sperm are stored in the epididymis for a period of time until mating occurs, and at that point they'll be passed through this whole reproductive tract here. And there's a couple of other glands that contribute some of the fluid that help move these sperm along, and these include the vas deferens and the prostate gland. So the ejaculate has some volume that is contributed by fluids from these 2 additional glands. Okay, and so according to the World Health Organization, typical sperm count for mammals would be on the order of 20 million sperm. So it's really a mind-boggling number of sperm that get transferred with every ejaculation, and one of those is hopefully going to be successful to fertilize the egg. So zooming in, this represents the testes, and in red here the portion of the tube known as the epididymis. And then zooming in further, you see the testes is actually jam-packed full of these small tubules called seminiferous tubules. And inside of the seminiferous tubules is where spermatogenesis is occurring. So these are all they're not really very big in diameter, but there's thousands of them, and they're really packed in there. And in the lab next week, you're going to get to see a section through a bull testes, and you'll see, I think it's just amazing and impressive to me how many of these tubules are inside the testes. Then looking at a cross-section for one of the tubules, we see this sort of arrangement where there's cells all along the periphery of the tubule, but the interior is a lumen. And so these sperm are being made in spermatogenesis close to the wall of the tubule, and then the mature sperm are released into the center lumen, and then all these tubules feed back together, and the sperm are collected in the epidermis. So here's just a cross-section that's been stained to sort of show you a little bit more of the morphology here. And all these long string-like things in the center are the sperm with their long tails, and the cells around the periphery are more circular in shape, and you can see some dark staining where DNA is condensing as it goes through meiosis or mitosis. And I guess this is an artist's depiction of what that would look like. So then if we zoom in even further and just consider one little section here from the wall to the lumen and the different cell types you would see, it looks like this. So the spermatogonia, the stem cells, they're most peripheral, located next to the wall of the seminiferous tubule. And then primary spermatocytes and secondary spermatocytes. So as they start to go through meiosis, they move further and further away from the wall. And in these sorts of sections, you may see cells with chromosomes at the metaphase plate or chromosomes in anaphase because they're going through meiosis. And then spermiogenesis is occurring where the cytoplasm is being sloughed off and other changes. And finally, the mature sperm released into the lumen of the tubule. Then we also have here in brown a couple of cells known as Sertoli cells. These don't make sperm, but they're present inside the seminiferous tubules, and they secrete some hormones and other substantive or nutritive properties to try to keep these stem cells happy and productive and dividing over the lifetime of the reproductive age of the male. Okay. So then in between the seminiferous tubules is another cell, and these are called interstitial cells or Leydig cells, and these are the cells that are producing testosterone, which obviously is an important male hormone, and so I just point these out as the source of testosterone within the testes itself. Okay, here's another section showing you spermatogonia and some of these different cell types with mature sperm in the middle, and an actual Sertoli cells that is inside the seminiferous tubule. Okay, let's look a little bit more at spermiogenesis and what's happening here. And so this is the process where we're converting these spermatids into mature spermatozoa. And I'm going to talk about 5 different things that happen. And even though I'm talking about them sort of one slide at a time here, you can take it that all of this is a continuous and overlapping and more or less simultaneous process that's occurring. So the first thing to point out is that the nucleus becomes very compact and actually basically shuts down transcription. So this serves a couple of purposes. For one thing, it makes the general size of the sperm head smaller, and that's just going to be more streamlined and perhaps better for the sperm that's trying to move quite a long distance within the female reproductive tract. Secondly, it's now non-transcriptional. If all these DNA is very compacted, it's somewhat protected as well, and there's not really a need for this cell to be doing a massive amount of transcription at this point. It has the structures that it needs. It needs to do a little bit of basic metabolism probably, but not any particular need to synthesize tons of enzymes or anything like that. So the nucleus becomes non-transcriptional. And so this is the drawing from your book, but the actual shape of these nuclei is quite a bit longer and thinner, and you can really detect the dark staining if you look at sections because this DNA is so compacted. Next thing is that we are going to collect a large number of mitochondria behind the nucleus. And mitochondria, as you recall from cell bio and other classes, these are the site where ATP is made. And this cell is going to need a lot of ATP because it's going to need to move that sperm tail and swim to the location of the egg. So there are many, many mitochondria, and they're all here in this sort of mid-region of the sperm, behind the sperm head, which contains the nucleus. Okay? The sperm also contain a centriole. So remember, the centriole is the microtubule organizing center. That's going to be important once we get to the fertilized egg, because that will set up one of the poles of the first mitotic spindle in that reconstituted diploid nucleus. And you can also make out all these mitochondria in sections because they're small and circular organelles and they're pretty easy to recognize, right there behind the highly condensed sperm nuclei. Okay, then importantly, the sperm make a flagellar tail, and this is going to be based on a core of microtubules. And so once again, the centriole, as the microtubule organizing center, is an important part of this because it initiates the growth of these really long microtubules, a whole bundle of them that extend to this flagellar tail. So in the tail, the microtubule core is going to be wrapped with this additional set of proteins called dense fibers because of the way that they appear in electron microscopy sections. And then the whole thing, the whole tail, is still surrounded by plasma membrane. Because this sperm is one cell, and so we have a plasma membrane that goes around the entire outside of that cell. So axoneme is the term that we use to describe the arrangement of microtubules and other proteins within the tail. And so if you just look at a cross-section of that tail, you see a number of microtubules. There's 2 in the middle, and there's 9 pairs that surround that. And it's a really characteristic arrangement for microtubules in flagella of different types. And it's even got a name, the 9+2 arrangement. And this seems to be